So, guys, as you know, for example, inside our bone, we have hematopoietic stem cells, guys. Hematopoietic stem cells. Can you just tell me the surface marker of? Oh, sorry, guys. We have hematopoietic stem cell. Can you guys just tell me the surface marker of hematopoietic stem cell?
CD34.
Okay, perfect. Okay, CD34. So, hematopoietic stem cells are basically divided into two broad categories or two broad lineages. One is our myeloid lineage and our lymphoid lineage. Just give me a second, sorry. Okay, so we have lymphoid lineage and myeloid lineage. So from the lymphoid lineage, we are going to see, okay, so in immunology, we will be mostly concerned with the lymphoid lineage. Which cells come out of the lymphoid lineage, guys?
Lymphocytes?
T cells.
Perfect. Your B cells and T cells.
T
Along
cells.
with that, natural killer cells as well. Okay. So the B cells are then transformed into plasma cells and they produce antibodies, right? The T cells are basically divided into two types, helper and killer, right?
Killer. Mm-hmm.
Okay. So can you just tell me the cells which are derived of your myeloid lineage, which we will be discussing in hematology? Okay,
RBC?
first one. Yes, erythrocytes.
Megakaryocytes.
Okay, perfect. Erythrocytes, RBC,
Megakaryocyte?
megakaryocytes. Perfect. Your
Platelets.
platelets, your monocytes, right?
Yes.
That are then
Neutrophils,
converted
eosinophils,
into
basophils.
Macrophages?
macrophages. Perfect. Then your basophils,
Neutrophils, eosinophils, basophils.
eosinophils, and basophils, right? So we will be discussing the myeloid lineage in detail once we start our hematology section. But here in this immunology section, we are concerned with lymphoid lineage, right? So now let's discuss thymus. Can you guys tell me the location of thymus, where the thymus is present? Hello?
Anterior mediastinum?
Yes, perfect. It's present in your interior mediastinum. Basically, it arises from your third pharyngeal pouch. So we have already discussed a condition, a pathological condition, in which we are going to see third and fourth pharyngeal pouches maldevelopment. What's that?
Digest syndrome.
DiGeorge syndrome. Okay, can you tell me that what's the function of the thymus?
Maturation?
Perfect. So T cells maturation. Okay, although the T cells and the B cells are formed in your bone marrow, but the B cells are released from your bone marrow in a mature state. But the T cells come out immaturely and then they are matured in your thymus, which we will be discussing each and everything, that how do the positive selection takes place and negative selection in order for the T cells to be matured, right? So, okay, can you guys tell me that we have a condition which is highly related with the thymus or a person having thymoma, very high yield. Like for example, yesterday we discussed small cell lung carcinoma is associated with Lambert-Eaton syndrome, right? That is pre pre synaptic. So what would be the antibodies that are post synaptic against acetylcholine? What's that condition called?
Myasthenia gravis?
Perfect. So myasthenia gravis is highly associated with thymoma or thymus. So those kind of individual who have myasthenia gravis, you must look for thymoma, because this is the thymoma which is producing these antibodies against the acetylcholine receptors. Now let's discuss another structure, lymphoid structure, that is our spleen, right? So basically what happens in the spleen, first of all, it is an arteriole, right? Okay. Surrounding this arteriole, you have a white kind of space present. Okay, that is basically known as okay white pulp. Surrounding the white pulp, you have your red pulp, right?
Yeah.
Hello. Okay. So basically, for this is your artery. So periarteriolar sheath. Somewhere here, your T cells are going to sit. Okay. And inside the white pulp, you are going to see formation of follicles. And whenever you see the term follicles, just remember you are going to see B cells there, which we will be discussing in details once we start discussing our lymph nodes, your primary follicles, secondary follicles. But remember, follicles, the B cells are going to sit inside your follicles, right? So, okay, then we have red pulp. Can you guys just tell me what's the function of your red pulp? It's quite easy. Sorry.
Allow cells to pass in and out.
Okay,
Old
it
RBC
destroys
removal?
your old RBCs.
Proliferation.
Destroy your old RBCs. Like there are certain sinusoids opening present between these red pulps. Like spleen is also known as graveyard of your red blood cells. Simply because the red blood cells, which like are old, are going to be destroyed inside your red pulp, right? Okay. Another thing related to spleen. Spleen is also going to produce certain antibodies. Like it's like consider that you half of your antibodies are made inside your spleen because spleen have B cells, right? And these B cells can produce antibodies, IgM, the first antibodies that come out of your B cells, right? Similarly, platelets can also be stored in your spleen. Okay. Hello.
Yeah, okay.
Hello. Okay. So now you guys are going to tell me. For example, if a person is having a spleen now, so he is at increased risk of which kind of infections? One of your you will answer me
Encapsulated?
very highly. Okay, perfect.
Encapsulated.
Encapsulated. So why is that so? Why do this person have increased risk of being infected by encapsulated organisms?
Because this is removed by spleen normally.
Okay, yes, normally it's removed by spleen, but secondly, remember whenever you have capsule around certain bug simply, so it cannot be directly eaten by your cells. That is the your first of all the antigen presenting cells are going to come, they are going to present them into the CD4 cells, right? But for example, if we have certain bugs who have this capsule, so you the antigen presenting cells cannot directly come and they cannot directly eat them and present them on their MSC class 2. You need a tag for them. That is IgM. You need IgM for the antigen presenting cells to recognize your encapsulated bacteria, right? So, for example, if a person is having a splenia, it means half of his IgM is are going to be
Yeah, your
absent.
voice is breaking.
So, in this case, your antigen presenting hello, 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 hello. Can you
Yes.
guys hear me? Hello.
Yeah, yeah.
Yes.
Okay.
Yeah.
Okay. So did you guys understand like for encapsulated organisms to be phagocytosed by your antigen presenting cells, we need antibody IgM, and we know half of the IgM is made by your spleen. So those individuals who do not have any spleen, it means there isn't this tag for the encapsulated bacteria, so that antigen presenting cells can recognize them. So simply he would be having infection with encapsulated bacteria. This is one of your U-World MCQs. They are going to test you on it. Okay. Secondly, spleen can be damaged during traumatic events, right? Like for example, we have an infection. If you guys remember, that is EBV infection. Remember, causing EBV infection caused which disease?
Burkitt lymphoma.
Okay, Burkitt lymphoma. Secondly,
Infectious mononucleosis.
Infectious mononucleosis.
Esophageal
Perfect.
carcinoma.
So in yes, nasopharyngeal carcinoma as well. So basically, those kind of individuals are like advised to not play contact sports because of damage to their spleens. Okay. Secondly, we have a condition that is known as hereditary spherocytosis. So what happens in hereditary spherocytosis is that normally your red blood cells are like biloid, like that, but it becomes sphere shaped. Why? Because your
Loss
spleen
of
is going to,
insulin spectrum.
yes, you have loss of your that is the like the structural component integrins and okay. So what happens as a result of that? These spherocytes can be eaten up by your spleen simply, or it can be destroyed inside the sinusoid. So for those individuals, basically what do? We just cut their spleen or remove their spleen because these spherocytes have good ability to transport oxygen, nutrients, each and everything. But the main issue is that spleen is going to destroy them. So you are going to see anemia. So that's why we simply remove their spleen, right? Hello.
Yes.
Okay. So now let's move towards secondary lymph node organ, that is our lymph node.
Dr. Ahmed?
Yes.
Hello. Dr. Ahmed, could you please repeat this hereditary spherocytosis concept once again, please?
Okay. We will be discussing it in like hematology in detail. But what happens in hereditary spherocytosis is that due to loss, like you have like inside the red blood cells, you have some supporting fibers simply. Okay. So if we have any mutation of those fibers, it means your red blood cells are now going to attain sphere shape round. Normally, your red blood cells are quite flexible to pass through these sinusoids and survive. But these sphere shaped red blood cells cannot survive in these sinusoids. So they will, okay, they are, although they are normal, their capability of transporting oxygen is normal. Their capability of undergoing normal function is normal. But the only problem is that they are going to be destroyed in your spleen sinusoids.
Okay.
So what's the treatment approach for them? Simply remove their spleen.
Okay. Got it.
Okay. Okay.
Thank you.
Okay. Welcome. Okay. Then you have lymph nodes. So as you guys know, for example, this is our lymph node. So you have basically two layers. One is your cortex and your guys. Okay. Medulla. Perfect. So basically, the cortex have certain areas that are known as follicles. Okay. Okay. We have two. Okay. We have discussed that B cells are going to be converted into plasma cells, right? And plasma cells are then going to produce your antibodies, right? Okay. So remember, we have two kind of follicles: primary follicles and secondary follicles. So in the primary follicles, you are going to see dormant B cells which aren't activated. So in the primary follicles, you are going to see dormant B cells. But in the secondary follicles, you are going to see activated form of these B cells. Okay? And secondary follicles is also known as germinal centers. Right? So it means, for example, if a person is having an infection, his B cells are activated. It means we are going to see germinal centers in his lymph nodes, right? Okay. Do you guys know what is isotype switching?
No.
Okay. The idea is that normally your B cells, whenever it converts into plasma cells, the first antibody which it releases is your IgM.
IgM.
ICM.
IgM. Okay. But if we want another antibodies, other antibodies, for example, if for the mucosal surfaces I need IgA, so it means I have to class switch. I have to make some more IgA. So for that, you are going to see interaction of certain receptors, which we will be discussing furtherly. So it means a conversion of antibodies from IgM into other forms. It's basically known as isotype switching. So can you guys tell me that isotype switching takes place in the primary follicles or secondary follicles?
Secondary follicle.
Secondary.
Secondary.
Perfect. Secondary follicles. Perfect. Okay, then you have an area that is known as paracortex. Like surrounding the cortex, you have paracortex. Can you just tell me which type of cells are present in your paracortex? One of your U World MCQ.
T cells.
Perfect. So T cells are going to sit in the paracortex, right? Okay. So you know whenever a person is having some viral or fungal infection, which we will be discussing, you are going to see mostly T cell response. Okay. We will be discussing that why are you going to see T cell response. But for example, if you have a question, a person is having some viral infection. So um, which zone of his lymph node is going to be enlarged or hypertrophied?
Paracortex.
Paracortical.
Paracortex. Perfect. It's one of your MCQ. Okay, so in the medulla, you are going to see the plasma cells, lymphocytes, and macrophages basically sitting in your medulla, right? Okay, then we have Peyer's patches. Do you guys remember what is Peyer's patches?
Lymphoid
Basically
aggregation
present in your
CGI.
ileum. Perfect. So basically, they are lymphoid tissues which are present in your GI, specifically the ileum, right? So you guys know that for your mucosal immunity, you need an antibody. What's that?
GA.
IgA.
IgA.
Perfect. Perfect. IgA. Okay. So what happens in Peyer's patches that, for example, inside the ileum you have Peyer's patches. Okay. So it means they are same as that of your follicular tissues, right? So it means they will be having germinal center as well. Hello. Okay. Can you guys see my screen?
No.
Okay. So it means your follicles are also going to be present in your Peyer's patches as well, right?
Yes.
Because inside the follicles we have B cells, and we need okay, B cells are going to be converted into plasma cells, and plasma cells are going to produce IgM further. Then we are going to undergo isotype switching, and then we are going to form IgA, which is important for your GI mucosal tract, right? Okay, so now let's discuss immunity types, that is innate immunity and adaptive immunity. So you guys are going to tell me about okay innate immunity. Okay, so can you guys tell me that innate immunity is specific or non-specific?
Non-specific.
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 Perfect. It's non-specific. It means, like for example, if a virus came, if a fungi came, for example, if a bacteria come inside your body, so they are going to coop out all of these three microorganisms in same way, right? It means they are non-specific. Simply, okay. Can you just tell me their speed? Would it be rapid or it takes some time?
Rapid.
Rapid.
Innate is rapid.
Perfect. It would be rapid, like it. Yes, it's rapid. Like it's your first line of defense. That is like present, hereditarily. So it means it would be rapid. Okay, can you guys just tell me that do they have memory kind or
No.
do
No,
we
No.
they
have
don't have.
what
No.
thing? Okay, so they do not have memory. Okay, what's the concept of memory? For example, if they do not have memory, it means whenever this organism, let's suppose this fungi, let's suppose attack next time, it means it would take same time for this reaction, right? Like it won't be fast the next time. The concept of memory is that if you are having memory, it means whenever you have second exposure to the same pathogen and you have memory against it, it means the second response would be exaggerated and rapid, right?
Right.
Okay, so the future response is basically equal to initial response.
Hmm.
Okay. Okay, so we are going to discuss can you just tell me the components of innate immune system?
Neutrophils,
Skin, mucous
macrophages,
membrane,
Okay, perfect. Complement macrophages, neutrophils.
neutrophils,
Okay,
monocytes.
yes, monocytes. So basically, they are going to undergo like a respiratory bust. If you guys remember the formation of free radical superoxide in order to destroy the organism. Okay. So innate immunity, it's basically it comes from your bone marrow, right? From the bone marrow, it increase right? Hello.
yes,
Eosinophils
Yes,
as
yes.
well, as we have watched in the myeloid lineage. Basophils, mast cells, monocytes, and natural killer cells. Okay, and the
Mm-hmm.
primary function for innate immunity is just replication and destroying it. It's basically effective against fungicides. Remember, eosinophils basically are against parasitic infections or helminth infection, right? Okay, and it's encoded germline. Like it, guys, can are you getting it? Hello,
Mm-hmm.
hello, guys, are you getting it?
No.
Sir, can you repeat? Can you repeat?
Your voice is getting cut.
Okay, hello, hello, can you guys hear me? Hello,
Mm,
hello,
yeah.
Hello.
So your voice was
Yeah, we
not
can.
clear
can you
previously.
guys?
Please
Can you repeat?
repeat.
Okay, okay, okay. Should I from where should I repeat?
Respiratory
The
busts and
primary
all.
response of the innate.
innate immunity cells,
Okay, so basically we were discussing about innate immunity, which is encoded germline. Like all of individual by birth, they are going to have innate immunity, right? Okay,
Mm-hmm.
and it's non-specific. Like for example, if we have like it do not like for example if we have three different bugs, it would like attack all of these bugs in same way. They do not have specific response to specific bugs simply as we have in adaptive immune system, which we will be discussing later. Similarly, the future response is like they do not have memory. If they do not have memory, it means the initial response is equal to the future response simply. Okay, and what are the component? You guys have just told me your eosinophils, your mast cells, your monocytes, your basophils, natural killer cells, right? This was all about.
Hello.
Hello.
Okay, is that clear?
Hello. Hello.
So we cannot hear you.
You guys hear me? Hello.
No.
Now we can hear.
So I don't know what's happening.
Hello.
Hello. Can you guys hear me? Hello. 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 Hello.
Yes, we can hear you
Yes.
now.
Okay, just give me a second, guys. Just give me a second. I'm just checking my internet connection. Just give me a second.
Yes.
Everybody.
Okay. So can you guys hear me well now? Hello.
Yes, yes.
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 Okay. Okay. So shall I repeat this in that immunity or not?
No. No, we
Okay.
got it.
So now let's discuss adaptive immune system. Okay. So
We can't see your screen.
can you guys tell me
It's bad again. It's bad again, Dr. Ahmed.
and antibodies have two regions, the variable region and constant region. And these variable regions are highly specific, which we will be discussing in detail. Like for example, let's suppose that I'm just giving you this for an example purposes. For example, if we have a bacteria who have one, two, three, this is for example their pattern. Let's suppose I'm just giving them a rough pattern. So it means, okay, we have three bugs, let's suppose. One is 1, 2, 3. I have just given them a random number. Other 3, 2, 1 and 2, 1, 3. It means that you have specific sites or epitopes for these bugs to attach to the B cells. It means that adaptive immune system is highly specific for a certain bug, right?
Right.
Okay. Like you only 2, 1, 3 variable site is going to attach to it. It's not going to attach to this or that simply. Okay, so it means that adaptive immune system is highly specific, which we'll be discussing in detail. You have different recombination, VDJ recombination, and all of those stuff. So don't worry about that. It's just an overview, right? Okay. So secondly, it's very slow. The speed of this pathway is quite slow. Why? Because we will be discussing that, for example, whenever a pathogen enter your bodies, first of all, your antigen presenting cells are going to, okay, let's study it here. You guys are going to tell me, for example, the pathogen came. First of all, your antigen presenting cells are going to eat them, right?
Yes.
Yes.
Okay, can you just tell me some of your antigen presenting cells, examples of some of your,
Dendritic cell,
okay, perfect.
macrophages,
Macrophages.
Dendritic cells,
B
macrophages,
cells.
B cells. Okay. So these antigen presenting cells are going to eat them. They are going to lyse them and they are going to present it on the one of their surface receptor. What's that?
MHC.
Which MSC class 1 or 2?
MHC class 2. Class 2.
Perfect. So it is going to present some of their components on MHC class 2, right? Then this antigen presenting cells is going to go where? It's going to present it to.
CD4.
CD.
Okay, perfect. So we have T cells. T cells, there are two types of T cells, helper and killer. What's the surface marker for helper cells?
CD4.
CD4. What's the surface marker for killer cells or cytotoxic cells?
CD8.
CD8 positive. Perfect. Your helper cells is furtherly divided into two types. Already discussed. T helper 1
TH1, TH2.
and TH2. So these antigen presenting cells are going to come, they are going to present these molecules, these foreign pathogen to CD4 cells, right? Via a T cell receptor. That is CD4 receptor, right?
Yeah.
Okay. Then what happened? For example, if it's an intracellular bug, which we have discussed in case of TB. Okay. So what do you think? TH1 response would be activated or TH2 response?
Each one.
TH1.
Perfect. Granuloma formation. So TH1 response would be activated. So this was the whole idea of, okay, so if we are having certain events, it means this response is quite slow. No?
Yes, yes.
Guys, please participate. Okay. So it means you are going to have a slow response, right? So you are also going to see formation of memory, which we will be discussing, that only peptide form of the pathogen are going to form memory, which we will be discussing the concept when we come to vaccination, right? So what are the component of your adaptive immune system response? Already discussed. Your T cells that are CD8 positive and CD4 positive. Okay, B cells, your plasma cells, and your immunoglobulins, right?
Right.
Yeah.
Okay, so you guys are going to tell me, we have discussed MHC class 2 receptor. Okay, what's MHC1?
Present on all cells on nucleated cells.
Perfect. So basically, MHC class 1 is present on all nucleated cells. Like, for example, this is one of my nucleated cell, let's suppose. So you are going to see MHC class 1 on its surface. So for example, if this nucleated cell is attacked by a virus that is intracellular, right? You know, virus is going to go inside your cell, right? So you are going to see presentation of some of the particles on MHC class 1. And which cells are going to recognize your MHC1?
CD8.
Perfect. CD8 positive cytotoxic, that is CD8 killer cells, are going to recognize these pattern on the surface of MHC1 and ultimately they are going to eat up all of this cell, right? Hello?
Yes.
Yeah.
Did you guys understand or shall I repeat it?
Repeat it.
Okay.
Yes, repeat, please.
Okay. So basically, MHC class 1 is present on all of your nucleated cells, right?
Okay.
Okay. MSC class 1. Okay, MSC class 2 is also only present on your antigen presenting cells, which we have already discussed. We will be doing it again and again. Don't worry about that. But it's just a general overview, right? So, MSC class 1 is present on all kind of nucleated cells, right? So, for example, if a fire, yes, okay. So, for example, if a nucleated cell is attacked by virus, you know what's the nature of virus? The virus simply take our protein making machinery and they just produce or viruses, right? Like for example, it would invade, it would just take our ribosomes, it would use our machinery in order to produce their own, like in order to reproduce their own self simply. Okay, so it means viruses are going to move into your cell, intracellular, they are intracellular, right?
Okay.
So okay, this is my nucleated cell. I have MHC1 on the surface of them, MHC1 on surface of them, right? So
Yeah.
for example, if this nucleated cell is attacked by a virus, so what would this cell do? This cell is going to take some component of viruses and it's going to present it on MHC1. Okay. Why do it present it on MSC1? This cell want to send signal to these cytotoxic cells that come and just kill me because I have virus growing inside me, right?
Yeah.
Okay. So cytotoxic CD8 positive T cell will come and it would attack. Like CD4 cannot come. CD4 is only going to bind to MSC class 2, which is present on your antigen presenting cells, right? Okay. Sometimes what happens, all of the bugs have virulence factor, right? And what virulence factor? In order to save themselves from our immune system, they have certain virulence factors, right? So one of the virulence factor of the virus can be that it do not let this cell to make MHC class 1 simply, right?
So, what is the difference between endogenous antigens and exogenous antigens?
Endogenous and
Exogenous antigens.
androgens.
Antigens.
Okay,
Endogenous and exogenous
okay,
antigens.
okay. Wait. Let me tell you. So endogenous are basically that those antigen which love to attack, which love to invade inside your cell. For example, in TB we have discussed, they love to go inside your macrophages, right? So inside the macrophages, they would just disrupt the formation of phagolysosomes to form, right?
Okay.
Okay, they invade inside the cell. So viruses are also endogenous, right? So sometimes once what happens, you have a virulence factor that disrupt the formation of MHC on this cell, right? Like MHC is also a receptor. This receptor is formed in your rough endoplasmic reticulum, right? And then it is being presented on the surface of your cell. So sometimes what does virus do? They do not let this MHC class 1 receptor to be present on the surface of nucleated cell. Like what do I mean? Like in this case, for example, if a virus just infect your cell and its virulence factor do not let MHC to come on its surface in order to save itself from CD8 positive cells, right? Okay.
Right.
So now what your natural killer cells are going to come. So natural killer cells would see all of those cells who do not have MHC class 1, very high yield. Natural killer cells are going to patrol around here and there. They would see those cells who do not have MHC, those nucleated cells who do not have MHC class 1, natural killer cells are going to destroy them. So this is the second mechanism our body coops out viruses, right? Is that clear? Or shall I repeat it again?
Clear.
Yes, who do not have MHC1. So NK cells would think that there is some virus inside your cell and its virulence factor isn't allowing MHC1 to be present on its surface. So it kills all of your cells, simply. So these are the two mechanisms which our body adopts in order to get rid of your intracellular or endogenous pathogen, that is your virus, right? We will be discussing it in detail again and again, but I'm just trying to make a core concept of you guys so that It's easy in the further lectures. Don't worry about that. We are going to repeat it.
Okay.
Okay. So this was all of the. So adaptive immune system. It means it's very slow. Okay. You are going to see memory, right? In case of adaptive immunity, right?
Yes.
And
Yes.
it's very highly specific, as I have already told you. For example, your antibodies, they have a specific pattern 1, 2, 3, 2, 3, 1. So they're highly specific towards pathogen, right?
Yes.
Okay, so now let's discuss antigen presenting cells. Again, antigen presenting cells. Okay, so antigen presenting cells can be your dendritic cells, right?
Mm-hmm.
Yeah.
Dendritic cells, your macrophages, your monocytes, and your B cells, right?
Yeah.
Yeah.
Antigen presenting cells are first of all phagocytose. They first of all phagocytose the pathogen, right?
Yeah.
Then they would just present some of its components on MHC class 1, right?
Mm-hmm.
Okay. Then what happens? MHC class 1 or 2?
Two.
Two.
2,
Two.
yes. Look, you guys are sleeping. Okay, MSC class 2. Then it's going to present it to helper cells or
CD4.
killer cells?
CD4.
CD4. They are helper cells or killer cells?
Helper.
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 Okay, helper cells. Then what happens? If it's an intracellular organism, let's suppose, which response am I going to see?
TH1.
TH1 response. And if it's extracellular or exogenous antigen, then
TH2.
yes, we are going to see TH2. Let me give you the concept of TH1 response and TH2 response. Okay, remember, very high yield. Those microorganisms or those bugs which invade inside the cell, you are going to see TH1 response or simply granuloma formation.
Mm.
Why? Because if you are seeing TH2 response is going to drive your B cells, remember,
Mm-hmm.
and your antibodies. The antibodies cannot enter your cell, right?
Mm-hmm.
Like for example, if I have IgG, can it enter my cell and destroy this pathogen?
No.
No. So those pathogens which are extracellularly, they can only attack or attack those kind of pathogens, right? So intracellular, only your macrophages are going to come. Either they can form a granuloma or either they can simply directly destroy all of the cells. So whenever you see an intracellular organism, you are going to see which response, TH1 or TH2?
TH1.
Perfect. Whenever we have an extracellular organism, which response, TH1 or TH2?
TH2.
And now you know why, because the antigens, the antibodies cannot enter your inside your cell in order to destroy that organ.
Sir, can you repeat intercellular and extracellular again?
Okay. So for example, if I have an organism that just enter inside your cell, for example, we have already discussed TB, let it grow inside your macrophages, right?
Yeah.
Alveolar macrophages. It's going to have a cord factor that's going to disrupt the formation of phagolysosomes to form. So I cannot have B cell response against them. Why? Because they are inside the cell. For example, if I'm having B cell response, it's converted into plasma cells. It have made IgG, let's suppose. So IgG cannot enter inside your cell in order to destroy the TB bug, right?
Yeah.
So, so it means I should activate my TH1 response in order to have macrophages and eat all of this cell, right? If it does not eat, either you are going to see granuloma formation, right? We have already discussed. And if it eats, simply it's going to destroy it. Okay. So now let's discuss B cells. B cells can also be your antigen presenting cells, right?
Yeah.
Okay. So B cells are first of all going to phagocytose. Then, guys, tell me.
Via MHC class 2 present to the
Yes,
CD4.
yes, it's going to present it on MHC class 2. Kindly, guys, repeat. Kindly participate. Then what happens? It's going to present it into which
CD4.
cell? CD4 cell. Okay. Okay, remember, guys. Very high yield concept, very high yield. Okay. So your T cells can only recognize those organism or those pathogen who are peptide in nature
Peptide or protein.
or proteinaceous in nature, right? Remember that, okay?
Hmm.
So we will be discussing the concept in vaccination, okay? So, so for example, when a B cell come in contact with CD4 cells, CD4, let's suppose, and your B cells. Now B cells are acting as antigen presenting cells, right? Right? MHC2. It must come to CD4. So we also have another co-stimulatory receptor. What's that? That CD40
CD40.
40
and
ligand.
CD40 ligand. So whenever this extra receptor on the T cells and B cells come in contact with one another, so they are going to drive the isotype switching. Okay, we have already discussed that the B cell, whenever it activates, it's going to convert into plasma cell. The first antibody which is going to release is IgM. But for example, if I need IgA, if I need IgG, if I need IgM, so it means I have to class switch it, right?
Yeah.
Okay. So how do I class switch it? It's going to come in contact with T cells, and whenever the CD40 and CD40 ligand co-receptors come in contact with one another, then you are going to see isotype switching.
Sir, can you repeat that the T cell only recognizes the peptide one?
Okay. 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 Just leave it now. Once we come to the vaccination, we are going to do it. Okay. Vaccination. Don't worry about it. Just leave it now. We are going to repeat it again and again. Don't worry about it. Now, are you clear that how do the isotype switching takes place?
Yeah.
Dr.
Yes.
El Future, can you just give us a quick overview of isotype switching? Okay. So can I say that CD40 ligand is very important for isotype switching?
Yeah.
Okay. It
Yes.
means that if I have any issue with CD40 ligand, I will only be producing IgM, right?
Right.
So this is one of
Yes.
your pathology which we will be discussing, that is hyper IgM syndrome. So what happens in hyper IgM syndrome? Simply you have defective CD40 ligand.
Mm-hmm.
Okay. So this was the idea. Okay, now let's discuss acute inflammation. So you guys are going to tell me, I think it's quite an easy topic, acute inflammation. What happens in acute inflammation?
Neutrophil first come.
Okay, that is neutrophil burst. Okay, let's, okay. First of all, what happens? You have a receptor present on the surface of neutrophils. What's the name of that?
I came on. I'm not sure.
Okay, toll-like receptor, remember?
Yeah.
Okay, this toll-like receptor is going to recognize certain patterns that are known as PAMs. What's PAMs?
Pathogen
Pathogen
associated
associated
molecular
Pathogen
molecular
patterns.
patterns.
associated molecular patterns. And can you just give me one of an example of PAMs? Perfect. And what is LPS?
LPS.
Lipopolysaccharide.
Lipopolysaccharide.
That's gram-negative bacteria have LPS, right?
In the cell wall. Mm-hmm.
That act as a endotoxin.
Endotoxin.
Can you just tell me what's the difference between endotoxin and exotoxin?
Exotoxin is released after the organism have been dead.
And exotoxin gram
Perfect.
positive.
Okay. So basically the idea is that endotoxin is basically normal component of this gram-negative bacteria cell wall, right? LPS. So whenever this is like this organism is split or it died, then it's going to release their LPS and it would act as endotoxin. And exotoxin is basically released from inside the gram-positive bacteria. So LPS is basically an endotoxin, right? So once your toll-like receptor is activated, you also need a co-receptor for that. What's that? That's CD40. Okay. So it's going to come, it's going to send some signals, and ultimately you are going to see formation of NFK beta. What's NFK beta? It's a nuclear
Nuclear
transcription
factor.
factor. Like it would go inside the nucleus and it would, okay, what's transcription? Guys, what's transcription?
Protein synthesis,
Yes,
transcription, and
like in protein
translation.
synthesis, first of all, we have transcription and then translation. In transcription, what happens? You are going to see a formation of messenger RNA, right?
Yeah.
Okay. So this transcription factor is going to go inside the nucleus and it's going to produce some specific kind of substances simply. That would be your cytokines, interleukins, and all of those stuff. And as a result of that, you are going to see an immune response being generated, right? Hello. Okay. So in acute inflammation, can you just tell me the cells which would first of all come to the site?
Neutrophils.
Okay, perfect. Neutrophils are going to come onto the side. Like there are certain factors that are known as chemotactic factors. What are chemotactic factors?
Interleukin 8.
IL-5.
Yes, interleukin
Interleukin
8.
5.
They are going to attract neutrophils towards the site of infection or inflammation. So for example, this is your blood vessel. So there are certain processes which are involved in the, like for example, here the infection is going on. So I need neutrophils to come to this side. So there are certain steps which are involved in the neutrophil migration. Can you guys tell me those steps?
Margination.
Okay, like first of all, you are going to push these neutrophils towards this margin, right?
Yeah.
If I want these neutrophils to come outward to the this side where the infection is going on, it means first of all I have to push these neutrophils towards peripheries, right? So that process is known as margination. Okay, in which you are going to see increased vascular permeability. Secondly, as they are moving, first of all I am going to slow their speed.
Rolling.
Yes, rolling. Like first of all they were moving like that, then I slowed their speed, so they started rolling. Okay. So can you just tell me that on the endothelial surface, which molecules or which substances are going to slow down the speed, or which selectins are involved in the process of rolling?
P.
Yes, P selectin and E selectin.
Selectin.
They are going to attach to silyl Lewis part of neutrophils. Like neutrophils have certain parts simply that is known as silyl Lewis. The P selectin and E selectin is going to attach to them and they are going to slow down their speed. Simply you can imagine of that I am adding some speed breakers here. Okay, so the P selectin and E selectin are simply the speed breakers. They would just slow down their speed and ultimately the neutrophils are going to roll down. They are going to start rolling, right? Okay, now I have slowed down the neutrophils. Thirdly, I should just stop the neutrophils on a single location. So I need some adhesion. Hello?
Chemotaxis?
Yes, it like all of this process is basically chemotaxis. There are certain, we are discussing each and every step by step. First of all, we are going to push neutrophils towards the periphery. Secondly, I'm going to slow down their speed. Just install some speed breakers or speed bumps. Hello?
Yes.
Okay. So they will start rolling. Fourthly, I'm going to stop them. How to stop them? I'm going to adhere them to the surface. So what's the molecules involved in the adherence of these neutrophils towards surface? That are ICAM. Okay, ICAM is going to stop them. Okay, neutrophils are going to attach to ICAM and they would just stop there. Now I need these neutrophils to move through this membrane and enter here to the site of infection, right?
Okay.
So that process is known as transmigration. Okay. So which molecules are involved in transmigration? That are PCAM. Simply. Okay. So let me repeat it. Let me give you an overview. First of all, for example, if a person is having acute inflammation, the neutrophils, how do the neutrophils activate? Okay, first of all, you are going to see toll-like receptor present on the surface of neutrophils. They are going to come in contact with pathogen-associated molecular patterns, right? They are going to send some transcription factors. These transcription factors are going to produce certain chemotaxic factors, certain cytokines, in order to move towards the site of infection simply. In acute inflammation, what happens? For example, if I need the neutrophils to come to this site of infection, that is outside somewhere in the tissue. So first of all, I would push the neutrophils towards the periphery. If I am pushing it towards the periphery, it means I am just arranging them on the margin. So this process is known as margination.
Okay.
Secondly, I'm slowing down their speed because they are like running in a like very fast. Okay, so I have to just install some speed bumps. There, those speed bumps are your P selectin and E selectin. Hello.
Okay. Okay.
Yes.
So they would just start rolling there. Now I want them to be on a single place or stay there. So then I'm going to adhere them. So ICAM is going to adhere them. Then I would just transmigrate them through this membrane. So this process is known as transmigration simply. Okay. So this was all about acute inflammation. Okay. Now let's discuss T cells. Okay. So before starting T cells, let's have an overview right from the beginning. Okay, quick overview. I'll be Dr. Reenu, kindly start with the hematopoietic stem cells. What are hematopoietic stem cells? Its lineages.
Okay, so the CD marker for the hematopoietic stem cell is CD34.
Perfect. You have
Okay,
two lineages. Okay, I'll be
two
asking you
lineage.
questions. Just answer those. Two
Okay,
lineages. Name them.
myeloid and lymphoid lineage.
From the myeloid lineage, which kind of cells are going to come out? Kindly,
The
guys, listen it. Okay.
From the myeloid lineage, the erythrocytes, the megakaryocytes, the monocytes, the basophils, eosinophils,
Perfect. From the lymphoid lineage.
B cells, T cells, NK cells.
Okay, perfect. Can you just tell me that about thymus? Where is thymus present and which pathology is associated with thymus?
Yeah, okay. So the thymus is located in the anterior mediastinum, and
Okay, perfect.
it is like derived from the third and fourth pharyngeal pouch,
Okay,
and
third
it,
pharyngeal pouch. Third
third
pharyngeal pouch.
pharyngeal pouch,
Okay,
and
can you just tell me the pathology associated with the absence of third and fourth pharyngeal pouch? What's the name of
uh,
that?
the George syndrome.
Perfect. Okay, what's the pathology associated with thymoma?
It's the myasthenia gravis.
Perfect. Okay, so here we are going to see maturation of which cells?
T cells.
Okay, maturation of T cells. Perfect. Dr. Alfutures, come forward. Dr. Alfutures. Okay, so you are going to discuss spleen. Can you just tell me that inside the spleen you have two regions? Can you just name them? Okay, perfect. White pulp. It is same as can I say that it's a secondary lymphoid tissue? Okay, so there would be T cells and B cells present inside them, right? Where are exactly the T cells location?
The
Periarteriolar sheath.
periarterial.
Okay, perfect. And B cells? Okay, in the germinal centers or follicles, primary follicles. The secondary follicle is your germinal center. Okay, okay. So, for example, if a person is having, okay, can you just tell me what's the other role of spleen? It's known as graveyard of what? Yes, it removes red blood cells. Okay,
RBCs.
okay, perfect. So they are also going to store platelets, right? And overall, half of the antibodies are made inside the spleen. That is IgM mainly. Okay, right? So, for example, if a person is having a splenia, why would he be prone to have encapsulated organism infections? Okay, perfect. So you need a tag molecule for the antigen presenting cells in order to recognize your encapsulated bacteria. Secondly, for example, if a person is having EBV infection, I would just tell him not to avoid contact sports. Why? Yes, because it's like quite okay. So secondly, in hereditary spherocytosis, what's the major problem and what's the treatment option? What do you think? Hello, doctor.
Mutation
Loss of bicconcave shape.
actin and spectide.
Okay, Dr. Maheen. Dr. Maheen. Dr. Maheen. In
Yes,
hereditary
there's a
spherocytosis, what's the issue?
RBCs loses their biconcave shape
Okay.
due to mutations in enchirion and spectrin
Yes, in
that results in spherules,
okay.
and these spherules will be destroyed by spleen.
So simply, what's the treatment option? Remove their spleen, right?
Splenectomy.
Okay. So, Dr. Ramesh, can you just tell us the different zones of your lymph nodes, and can you just tell me the type of cells associated with them?
Uh, one folliculars.
Okay, we have cortex.
Yes.
Then we have medulla. Inside the cortex, we have follicles, right?
Yes.
Then, okay.
Mm,
Anyone else? Dr. L Futures. Okay, the follicles are basically of two types, primary and secondary. Can you just tell me in primary follicles which kind of B cells are present, activated or inactivated? Or dormant? In
Inactivated.
secondary, okay, secondary follicles. Okay, and they are also known as germinal centers, right? Okay, in the Pyer patches, I also have some extra nodal tissues, right? And they have follicular cells as well, in which these cells are going to sit. They are going to produce which antibody mainly?
IgA.
IgA, perfect. Okay, Dr. Remis, can you just tell us about innate immune system, innate immunity? Is it specific or non-specific?
Non-specific.
Is it rapid or slow?
Rapid.
Do they have memory or no?
No.
No. Okay. Would the future response similar to that of initial response?
Similar.
Okay.
Yes.
And which can you just tell me the the associated cells which are in innate immune system?
Neutrophils.
Okay, perfect.
Eosinophils.
Eosinophils, basophils. Perfect.
Mast cell.
So,
Natural killers.
okay, now doctor. Okay, okay. Who want to just volunteer? Antigen. Okay, Doctor Futures. What are antigen presenting cells? Tell us some types of antigen presenting cells. Okay, okay, they are going to eat up the pathogen. Then, what's the second step?
After it eats it up, then it will present it to the MHC class.
Class,
Class
class
one.
one or two?
One.
Class two.
Class
Class
two.
one is present on your nucleated cells.
Yeah, class two.
This is one of your UWorld MCQ. They are going to ask you antigen presenting cells have class 1 or class 2. It's class 2. Then what happens?
Then it will CD4 will come in.
Okay, CD4 is going to come in contact with MSC. Then, for example, if I have intracellular, okay, extracellular organism, would it be TH1 response or okay, perfect, TH2 response. TH2 response is going to drive,
IgG production.
okay, B cell response. Perfect. So, for, okay, can you just tell me that MSC class 1 is present on which kind of cells?
Nucleated.
Which,
One query just. You were saying extracellular TS2, intercellular TH1, right?
yes.
Okay, sir.
Yes.
Okay. So, can you just tell us that, for example, if a person is missing with MSC1, which cells are going to destroy
Natural
the
killer
complete?
cells.
NK cells.
Perfect. NK cells. Okay. So, okay, antigen presenting cells. Okay, MSC1, 2. Okay, can you just tell me the, okay, Dr. Remis, can you just tell me the um, phases or the steps associated in acute inflammation?
Detection of PAMP, pathogen associated molecular pattern by
Okay.
toll-like receptors, and
Okay.
then it will release NF kappa beta, which will
Okay.
lead the neutrophils to produce certain chemotactic agents that will help in migration, and then rolling, and then adhesion, and then transmigration.
Okay, perfect. So it was all about, okay. So now let's discuss T cells. Okay, we'll be doing it in detail again and again. Don't worry, guys. We'll be repeating it again and again. So, and the best way to have grip on your immunology is to discuss it again and again. So, discussion is very important, right? So, you guys must participate in discussion. Even if you don't know, so we are here to help you. Okay, we will help each other. Don't worry about that. So, can you just tell me that T cells are made where?
Bone marrow.
No, they're made in the bone.
Okay,
Mesenchymal
futures, where are they made?
Made in bone marrow.
bone.
Okay, they
Both
are
are
made in bone marrow and they are matured in thymus.
time.
Mm-hmm.
Okay.
Yeah, I
Okay,
remember
then
it as like B for B and T for T.
yes, perfect. So B cells made in bone marrow and matured in bone marrow. T cells are known as T cells because they are matured in thymus.
Yeah.
Okay, so can you just tell me the surface markers of T cells, the helpers and the killer?
CD4 and CD8.
CD4 and CD8 positive. Okay, so now let's discuss how do the maturation takes place in thymus. For example, this is your thymus.
Sir, please give us 5 minutes break.
Okay, you need 5 minutes of break.
Not really.
Okay, okay, let's have 5 minutes of break. Then we are going to come and then we are going to finish it. Are you guys enjoying or not?
Yeah,
Yeah.
very much.
Yes. Yes.
Yes.
Sir, what is it like? Till how much time is there? Like, how much time will it take for the class?
Okay, we are going to have 2 hours of class. Yesterday I already discussed. We have 3 days. We are going to repeat it again and again, again and again. Don't worry about that. Once you leave the class, I guarantee you each and every one you would have complete knowledge of immunology. Don't worry about that. Okay. So let's have
Okay.
5 minutes of break. Then we are going to begin. Okay.
Okay.